alterations for malignant mesothelioma. We have obviously a really good understanding of the genetics of lung adenocarcinoma, also now of lung squamous tumors, even of head and neck tumors. For mesothelioma, this work is still ongoing, and I'll show you a first flavor of this. The Cancer Genome Atlas project is actually currently working on a data set of about, I think, 88 tumors. This is a first glimpse at a cohort that was actually done in Chicago, but I do believe that there's a lot of overlap. These are my disclosures. None of them are relevant for this talk. This is in the morning. I don't know. Anybody knows where this is? It's just around the corner. When you walk this direction. So, I think it's a beautiful setting. So, we all know that mesothelioma is relatively uncommon, and henceforth the genetic aberrations, the genetic background has been incompletely understood. There were a number of genetic aberrations that have been reported using single gene or very limited multiplex panels. We have known now for about 2 or 3 years that BAP1 seems to be a germline or somatic likely tumor suppressor aberration. There have been reports of EGFR mutation, especially in peritoneal mesothelioma. However, in our own experience, not very common and unclear how targetable. In my mind, it echoes a little bit the discovery of EGFR mutations in glioblastoma, and even in head and neck or squamous tumors. It seems like there are EGFR mutations, but they're not in the hotspots in the 2K domain, and the therapeutic implications are unclear. So for EGFR in mesothelioma, there were suggestions of TK domain mutations, and there was even a trial for this, but it definitely isn't the home run that people were thinking. NF2 loss was reported by Sloan Kettering, and then also others, loss of P16. But the key questions really are, if you do this more comprehensively, what is the incidence of potentially targetable genetic aberrations, and can we potentially subdivide tumors into categories that might either be prognostically or therapeutically important? So in this study, we picked about 60 patients that were treated at the University of Chicago, where we had tissue, and we used a panel, a next-generation sequencing panel, to get an overview. And this is a very high-grade quality, clear quality assay to identify genetic aberrations and then correlate those with clinical outcomes. So this is an overview of what we did. These were usually surgical specimens. We extracted DNA. This was a capture-based enrichment sequencing on the Illumina platform, and then bioinformatic analysis. As I mentioned, about 60 malignant mesotheliomas, 57 all treated at the University of Chicago. We used this panel. And I think the quality of the data is probably the most important to highlight. This is clear grade, and both mutations, copy number aberrations are very reliable in this study. We also did some limited, bioinformatic analysis, trying to identify the dignity of these mutations. You can identify a mutation. On the other hand, the prediction of whether it is actually targetable or potentially, clinically meaningful or biologically meaningful is a different question. Is it a driver or a passenger? And these prediction algorithms, they're good, but they're definitely not currently good enough to really actually use clinically. This is a gene list. I don't want to go into too many details. But essentially, this is a bioinformatic tool that I just mentioned. There's really different level of evidence to identify a driver character. There's a tool developed at Johns Hopkins by the group around Rachel Karchen called CHASM, which really is intended for cancer samples to identify drivers. It isn't very sensitive, but it's reasonably specific. So I think if you do have a CHASM score that is suggestive of a driver, it is oftentimes meaningful. You can just look for recurrence patterns. Statistically enriched genetic aberrations usually are selected for, meaning it's important. Prior knowledge, if you find the same mutation, BRAF V600E, oftentimes that's very meaningful. And then ultimately we need validation, be it drug sensitivity, or the gold standard would be obviously in patients with clinical trials. So we did these types. So 72% were pleural tumors, 28% peritoneal. Most of these were epithelioid. We had a good number of biphasics. We actually had no tumors that were truly purely sarcomatoid, which in part may be, the selection bias of what we see. Very high sequencing depths, larger 500x, given that this was a panel approach, which is different, obviously, when you do exome sequencing, where you get more genes covered, but at less depth. So this is actually an overview of the results. You can see here, BAP1 at the very top is altered in the vast majority of tumors. This is higher than what has been previously reported. You can also see the different mechanisms. I don't know how well the colors come out here, truncations, homozygous deletions. It is very interesting that BAP1 is so prominent and that there's really a mix of different aberrations. There's even a rearrangement, although this is an inactivating rearrangement here shown in yellow. It seems like, unlike other tumor suppressor genes where usually there's a preferred mechanism of genetic inactivation for malignant pleural mesothelioma for BAP1, it seems like any mechanism is acceptable as long as it leads to inactivation. Analogous to the Sloan Kettering data, NF2 is commonly altered. It's the second most altered gene. P16, CDKN2A, CDKN2B comes along for the right. And those are all tumor suppressors. So the top aberrations for MPM are tumor suppressors, really. Let me see how I get this. Yeah. So common aberrations in tumor suppressors: 50% BAP1, 26% NF2, P16 and 25% a quarter, and then P53 is relatively uncommon at about 10%. When we talk about the other part that I think is oftentimes important is when we talk about mutual exclusivity. We know this, for example, in lung adenocarcinomas. Oftentimes that's meaningful. It is not a perfect mutual exclusivity, but we do see that TP53, P53, P16, and ZD2 are mutually exclusive. So here you can see they never occur at the same time. And that probably is meaningful. A tumor seems to require one or the other, but not any combination. And I think that, again, is, I think, a sign of validity that's important. The same is true also for NF2 and TP53. So just to provide you an overview, again, BAP1 has been described previously as a germline aberration, really a key aberration for malignant mesothelioma, with 50% of tumors having this with a very high fidelity assay. You can see here a number of genetic aberrations. As I mentioned, we have this interesting pattern. We don't know if this is targetable. In our case here, we actually don't know how many of these are germline versus somatic. We believe that almost all of them are somatic. There are prediction algorithms, but we're not 100% confident, as this is an assay that does not usually sequence normal, and we've only done this for a few cases for validation. NF2, I mentioned, is this potentially targetable? There were suggestions of that in the past. A link of NF2 to the PI3K, AKT, mTOR pathway. In my experience, that has not been borne out. PI3K inhibitor trials have some activity, but there has been, as far as I know, no really good relationship with NF2 status. Maybe other things such as DNA PK may be more beneficial, but really the link of NF2 to targetability is still unclear. P16, we know one of the most commonly altered tumor suppressors in many, many other tumor types. Also here, unclear if targetable, but maybe we could target with CDK inhibitors, which are currently in development, and that's something we should keep in mind. And then finally, what we really wanted to see is there a genetic difference between peritoneal and epithelial tumors? And at least in our study, which is a relatively small sample size, we did not see genetic differences. And that's obviously very intriguing. Genetically, they seem to be identical. In my mind, it's almost like, in other tumors like lung cancer, the difference between mesenchymal and epithelial differentiation. And I think that's important potentially clinically, but we don't fully understand. It seems more that there are other mechanisms that relate to that. And part of that could also be that our pan is limited and that there's other mutations that actually go along with that. The other part I wanted to mention is when we talk about something that's potentially against targetable, we did find some recurrent BRCA2 mutations as well as ATM aberrations. This is a DNA repair signal. It's only 10%, but again, 10% might be good enough, at least for some patients, if we identify those, to consider DNA damaging related agents such as PARP inhibitors. So finally, when we really talk about oncogenes, there's very little signal for oncogenes. You know, people have reported aberrations in the PI3K pathway. We only found one single mutation in PIK3CA. Granted, this was a canonical mutation and was predicted as a driver, but that's really uncommon. And as you know, PI3K mutations are not good predictors of benefit from PI3K inhibitors. We found one RAS mutation, again, a driver. We did find one EGFR mutation, and using a prediction tool, it was predicted to not to be a driver and has not been reported for lung cancer to be actually amenable. And finally, we did find something that were interesting. TSC2 has been discussed as a rare but potentially targetable aberration with mTOR inhibitors, and we did think that that was important. There were two cases actually, you know, PTEN, INPP4B, where other aberrations, again, may be relevant, but nothing obviously that we should have as a large target that we should develop. So the other part I wanted to mention was exactly these oncogenes. There are other kinases such as EphA3, again, the PI3K pathway, EGFR, just to show you where they stand, and then potentially STK11 and MDM2. But really, all of these are really uncommon. This was a case report from Dana-Farber, published by Jochen Lorsch and his group, looking at everolimus in a patient with a TSC2 mutation with a dramatic response. So again, even rare aberrations may be meaningful, but we have to look for them. The final part, so this is an overview of what I just talked about, mostly a tumor suppressor disease. The last part that we did genetically was we actually did compare at the gene expression level epithelial tumors with biphasic tumors, and they are clear different, and that's obviously not surprising, you know, cell cycle, DNA replication. But what I thought was very interesting was that there's this immune therapy signal of cytokine signaling, toll-like receptors, and that's something that I think needs to be explored further. We already know that immunotherapy has activity in mesothelioma, and you know, just related to this, we actually did some additional analysis, you know, looking at this pattern of inflammation in mesotheliomas. And just like what we see in lung cancer, in melanoma, in head and neck tumors, we do see a signal of inflammation of tumor that are inflamed with a T cell or a cell inflamed phenotype. And you can see a number of checkpoints are expressed, including PD-L1, TIM3, LAG3, Tregs, macrophages. And obviously, as you know, macrophages are very prominent in mesothelioma. So I think those are potentially targets. If you cannot target oncogenes, maybe we should have this signal for immunotherapy. And just to show you a little bit more, we also did further analysis with multiplex color imaging, again suggesting that immunotherapy might be very interesting and prominent. Interestingly, PD-L1 expression in these tumors was mostly tumor-driven and not driven by immune cells, and was actually independent of CD8 infiltration, which is different from lung cancer. And with that, I would like to finish. We have a trial that's currently ongoing using a checkpoint inhibitor. We're actually done with the first stage, and the second stage will enroll soon. And so I would like to conclude that mesothelioma is a disease of tumor suppressors. The common ones are BAP1, 50%, and then 25% each of those categories. Oncogene aberrations are rare. Unlike prior reports, neither PIK3CA nor EGFR mutations were common, and that includes even in pleural tumors. So this one EGFR mutation did occur in a peritoneal tumor. Targetable aberrations do occur, but they're quite rare, and especially in a rare disease, that's difficult to develop. And finally, we still don't understand the genetic differences between the different histologies. Further work needs to be done. Again, I would like to point to the great effort by the Cancer Genome Atlas project, which I think will have a readout probably next year. And finally, I showed you the immune signal that I think is really prominent and maybe the one best option we have to target these tumors. So finally, I'd like to acknowledge the team at the University of Chicago, the amazing mesothelioma team, led by Dr. Hattie Kindler. We have great support in pathology, in surgery, in radiation, in radiology, and then also my collaborators, and I came out of Ravi Salgia's lab, my own lab, as well as the people who collaborated, such as Alia Hussein, and the people at Jounce Therapeutics who did the immunofluorescence staining. Thank you so much for your attention.
John?
Just a quick question, because we've brought it up a lot during this meeting, but the basically aberrations in the PI3 kinase AKT/mTOR pathway, and we see them a lot, and we keep saying the same thing, which is, yeah, but targeting doesn't seem to be. That pathway is a master switch. It's actually cells cannot survive without it, which means there's definitely always redundancies, and I think that's what we're seeing is that switch alone. Should we get away from trying to target that alone and target it always with something else? Because again, it's just itself, we're gonna bypass that pathway. It just the cell has to have ways around it. And should we be really thinking about what we should be adding with the PI3 kinase inhibitor in those cells? And then how do we figure out what that something else is?
I think it's a great question. We see PI3K mutations all across cancer, and it does correlate. Mutations do correlate with more benefit, but it's not a strong correlation. It's nothing like lung cancer, EGFR mutations, ALK aberrations. So my suspicion is that, there's flavors of that. You can look at canonical PI3K mutations. It seems like there's difference between, the kinase dominant helical, so a 1047 may be more predictive than a 542, as I showed you, but none of these are great or good enough. And then there's atypical mutations where we really don't know what it means. My suspicion is, one way could be maybe we need that second pathway that's important, and nobody has figured out what that actually is. I also wonder if there's interplay with other things. Maybe the function is not as a pure driver the way that we see EGFR. Maybe it has something to do with the immune system. Maybe it has something to do with, the way the tumor grows. And those type of studies haven't been done. I think people just keep this very simplistic rationale of, there's an oncogene that's altered, we target it, the tumor has to die. But maybe we change the history of the disease, and nobody has really done a large enough study to do this. I don't think we need to figure this out in mesothelioma. I think we can figure this out probably in other cancer types. And finally, the other problem is we sequence DNA. That means we miss half the story. And Dave Carbone would say we probably also should do proteomics. So I think we have to become more comprehensive. I am a big believer that profiling and collecting information will eventually lead to results. Even if it's not actionable, we have to do this on trials with patients where we have outcomes so we can actually interpret the results and identify biologic subgroups. So, I don't have an answer for your question, but I think the fact that these are common means that they're meaningful. Yeah.
Jerry?
Thanks for a great talk. I imagine the majority of the patients had clear asbestos exposure. Did you have a cohort that didn't, and did you see any patterns there?
I'm sorry, had exposure to?
Asbestos.
Yes.
And those that didn't?
Yeah, so we have asbestos data, which was read out by the pathologist. We have not done the correlation. So I don't have any interpretation. My initial impression, just looking at the slides or looking at this myself, is that it's going to be a messy correlation, and the numbers are too small to make that correlation. I think that the Cancer Genome Atlas project, which has more tumors, it's only about 90 tumors, but there I think the criteria for inclusion were a lot more stringent. They wanted tumors that did not have, talcum pleurodesis, and a number of other factors that might give us a clearer signal. So I actually hope that the TCGA cohort that I think will come out soon will give us a better answer there. Yeah.
Did you see any differences between the peritoneal and pleural mesotheliomas in terms of the common mutations?
We did not. So I didn't mention this, but we did not. We oftentimes say they're different diseases. We didn't see a big difference. We didn't see a difference between epithelioid and sarcomatoid. We also did not see a difference between peritoneal and pleural. And I always hear that biologic or clinically they are different. They behave differently. And then there's this pattern or the suggestion of EGFR mutation, but we didn't see it. So maybe we looked at the wrong genes, but I have a suspicion that there is clearly more similarities than differences. Again, I think the question of asbestos might be important because some of these might be less asbestos exposed. So I think there probably are differences, but we just didn't have the granularity to find it.
Great. Thank you.
Sure.